nucleus. The nucleus constitutes the central, more dense, part of the cell. It is usually rounded or ellipsoid. Occasionally it may be elongated, indented, or lobed. It is usually 4 to 10 pm in diameter. The nucleus contains inherited information that is necessary for directing the activities of the cell, as we shall see below. In usual classroom slides stained with haematoxylin and eosin, the nucleus stains dark purple or blue while the cytoplasm is usually stained pink. In some cells the nuclei are relatively large and light staining. Such nuclei appear to be made up of a delicate network of fibers. The material making up the fibers of the network is called chromatin, because of its affinity for dyes. At some places, in the nucleus, the chromatin is seen in the form of irregular dark masses that are called heterochromatin. At other places the network is loose and stains lightly. The chromatin of such areas is referred to as euchromatin. Nuclei which are large and in which relatively large areas of euchromatin can be seen are referred to as open-face NOC/EI. Nuclei that are made up mainly of heterochromatin are referred to as closed-face nuclei, Fig 1.33. In addition to the masses of heterochromatin, which are irregular in outline, the nucleus shows one or more rounded, dark staining bodies CAIE nucleoli, see below. The nucleus also contains various small granules, fibers, and vesicles, of obscure function. The spaces between the various constituents of the nucleus described above are filled by a base called the nucleoplasm. With the the nucleus is seen to be surrounded by a double-layered nuclear membrane or nuclear envelope. The outer nuclear membrane is continuous with endoplasmic reticulum. The space between the inner and outer membranes is the perinuclear space. This is continuous with the lumen of rough ER. The inner layer of the nuclear membrane provides attachment to the ends of chromosomes, see below. Deep to the inner membrane there is a layer containing proteins and a network of filaments. This layer is called the nuclear lamina. Specific proteins present in the inner nuclear membrane give attachment to filamentous proteins of the nuclear lamina. These proteins, called lamins, form a scaffolding that maintains the spherical shape of the nucleus. At several points the inner and outer layers of the nuclear membrane fuse, leaving gaps called nuclear pores. Each pore is surrounded by dense protein arranged in the form of 8 complexes. These proteins and the pore together form the pore complex. Nuclear pores represent sites at which substances can pass from the nucleus to the cytoplasm and vice versa, Fig 1.19. The nuclear pore is about 80 nanometers across. It is partly covered by a diaphragm that allows passage only to particles less than 9 nanometers in diameter. A typical nucleus has 3,000 to 4,000 pores. It is believed that pore complexes actively transport some proteins into the nucleus, and ribosomes out of the nucleus. Nature and significance of chromatin. In recent years there has been a considerable advance in our knowledge of the structure and significance of chromatin. It is made up of a substance called deoxyribonucleic acid, usually abbreviated to DNA, and of proteins. The structure of DNA is described on page 31. It is in the form of a long chain of nucleotides. Most of the proteins in chromatin are histones. Some non-histone proteins are also present. Filaments of DNA form coils around histone complexes. The structure formed by a histone complex and the DNA fiber coiled around it is called a NOC/eosin. Nucleosomes are attached to one another forming long chains, Fig 1.34. These chains are coiled on themselves, in a helical manner, to form filaments 30 nanometers in diameter. These filaments constitute chromatin. Filaments of chromatin are again coiled on themselves, supercoiling, and this coiling is repeated several times. Each coiling produces a thicker filament. In this way a filament of DNA that is originally 50 mm long can be reduced to a chromosome only 5 pm in length. A little calculation will show that this represents a reduction in length of 10,000 times. Some details of the formation of a histone complex are shown in Fig 1.35. 5 types of histones are recognized. These are H1, H2A, H2B, H3 and H4. Two molecules each of H2A, H2B, H3 and H4 join to form a granular mass, the nucleosome core. The DNA filament is wound twice around this core, the whole complex forming a nucleosome. The length of the DNA filament in one nucleosome contains 146 nucleotide pairs. One nucleosome is connected to the next by a short length of linker DNA. Linker DNA is made up of about 50 nucleotide pairs. Heterochromatin represents areas where chromatin fibers are tightly coiled on themselves forming solid masses. In contrast, euchromatin represents areas where coiling is not so marked. During cell division, the entire chromatin within the nucleus becomes very tightly coiled and takes on the appearance of a number of short, thick, rod-like structures called chromosomes. Chromosomes are made up of DNA and proteins. Proteins stabilize the structure of chromosomes. Nucleoli. We have seen that nuclei contain one or more nucleoli. These are spherical and about 1 to 3 pm in diameter. They stain intensely both with haematoxylin and eosin, the latter giving them a slight reddish tinge. In ordinary preparations they can be distinguished from heterochromatin by their rounded shape. In contrast, masses of heterochromatin are very irregular. Nucleoli are larger and more distinct in cells that are metabolically active. Using histochemical procedures that distinguish between DNA and RNA, it is seen that the nucleoli have a high RNA content. With the M nucleoli are seen to have a central filamentous zone, pars elementosa, and an outer granular zone, pars granulosa, both of which are embedded in an amorphous material, pars amorphosa, Fig 1.36. Nucleoli are formed in relationship to the secondary constrictions of specific chromosomes. These regions are considered to be nucleolar organizing centers. Parts of the chromosomes located within nucleoli constitute the pars chromosoma of nucleoli. Nucleoli are sites where ribosomal RNA is synthesized. The templates for the synthesis are located on the related chromosomes. Ribosomal RNA is at first in the form of long fibers that constitute the fibrous zone of nucleoli. It is then broken up into smaller pieces, ribosomal subunits, that constitute the granular zone. Finally, this RNA leaves the nucleolus, passes through a nuclear pore, and enters the cytoplasm where it takes part in protein synthesis chromosomes. Haploid and diploid chromosomes. We have seen that during cell division the chromatin network in the nucleus becomes condensed into a number of thread-like or rod-like structures called chromosomes. The number of chromosomes in each cell is fixed for a given species, and in man it is 46. This is referred to as the diploid number, diploid equals double. However, in spermatozoa and in ova the number is only half the diploid number, i.e., 23, this is called the haploid number, haploid equals half. Autosomes and sex chromosomes. The 46 chromosomes in each cell can again be divided into 44 autosomes and 2 sex chromosomes. The sex chromosomes may be of two kinds, X or Y. In a man there are 44 autosomes, 1 chromosome, and 1 achromosome, while in a woman there are 44 autosomes and 2 X chromosomes in each cell. When we study the 44 autosomes we find that they really consist of 22 pairs, the 2 chromosomes forming a pair being exactly alike, homologous chromosomes. In a woman the 2 X chromosomes form another such pair, but in a man this pair is represented by 1 X and 1 Y chromosome. We shall see later that 1 chromosome of each pair is obtained, by each individual, from the mother, and 1 from the father. As the 2 sex chromosomes of a female are similar, the female sex is described as homogametic, in contrast the male sex is heterogametic. Significance of chromosomes. Each cell of the body contains within itself a store of information that has been inherited from precursor cells. This information, which is necessary for the proper functioning of the cell, is stored in chromatin. Each chromosome bears on itself a very large number of functional segments that are called genes. Genes represent units of stored information which guide the performance of particular cellular functions, which may in turn lead to the development of particular features of an individual or of a species. Recent researches have told us a great deal about the way in which chromosomes and genes store and use information. The nature and functions of a cell depend on the proteins synthesized by it. Proteins are the most important constituents of our body. They make up the greater part of each cell and of intercellular substances. Enzymes, hormones, and antibodies are also proteins. It is, therefore, not surprising that one cell differs from another because of the differences in the proteins that constitute it. Individuals and species also owe their distinctive characters to their proteins. We now know that chromosomes control the development and functioning of cells by determining what type of proteins will be synthesized within them. Chromosomes are made up predominantly of a nucleic acid called deoxyribonucleic acid, or DNA, and all information is stored in molecules of this substance. When the need arises, this information is used to direct the activities of the cell by synthesizing appropriate proteins. To understand how this becomes possible, we must consider the structure of DNA in some detail. Basic structure of DNA. DNA in a chromosome is in the form of very fine fibers. If we look at one such fiber, it has the appearance shown in Fig 1.37. It is seen that each fiber consists of two strands that are twisted spirally to form what is called a double helix. The two strands are linked to each other at regular intervals. Note the dimensions shown in Fig 1.37. Each strand of the DNA fiber consists of a chain of nucleotides. Each nucleotide consists of a sugar, deoxyribose, a molecule of phosphate and a base, Fig 1.38. The phosphate of one nucleotide is linked to the sugar of the next nucleotide, Fig 1.39. The base that is attached to the sugar a molecule MIB adenine, guanine, cytosine, or thymine. The two strands of a DNA fiber are joined together by the linkage of a base on one strand with a base on the opposite strand, Fig 1.40. This linkage is peculiar in that adenine on one strand is always linked to thymine on the other strand, while cytosine is always linked to guanine. Thus the two strands are complementary and the arrangement of bases on one strand can be predicted from the other. The order in which these four bases are arranged along the length of a strand of DNA determines the nature of protein that can be synthesized under its influence. Every protein is made up of a series of amino acids, the nature of the protein depending upon the amino acids present, and the sequence in which they are arranged. Amino acids may be obtained from food or may be synthesized within the cell. Under the influence of DNA, these amino acids are linked together in a particular sequence to form proteins. Further details of DNA structure. In the preceding paragraphs, the structure of DNA has been described in the simplest possible terms. We will now consider some details. The structure of the sugar deoxyribose is shown in Fig 1.42. Note that there are 5 carbon atoms, and also note how they are numbered. Next observe, in Fig 1.43, that C3 of one sugar molecule is linked to C5 of the next molecule through a phosphate linkage, P. It follows that each strand of DNA has a 5 end and a 3 end. Next observe that although the two chains forming DNA are similar, they are arranged in opposite directions. In Fig 1.43, the 5 end of the left chain, and the 3 end of the right chain lie at the upper end of the figure. The two chains of nucleotides are, therefore, said to be antiparallel. The C1 carbon of deoxyribose give attachment to a base. This base is attached to a base of the opposite chain as already described. The reason why adenine on one strand is always linked to thymine on the other strand is that the structure of these two molecules is complementary and hydrogen bonds are easily formed between them. The same is true for cytosine and guanine. Ribonucleic acid, RNA, in addition to DNA, cells contain another important nucleic acid called ribonucleic acid or RNA. The structure of a molecule of RNA corresponds fairly closely to that of one strand of a DNA molecule, with the following important differences. A. RNA contains the sugar ribose instead of deoxyribose. B. Instead of the base thymine it contains uracil. RNA is present both in the nucleus and in the cytoplasm of a cell. It is present in 3 mn forms, namely messenger RNA, mRNA, transferRNA, tRNA, and ribosomal RNA. Messenger RNA acts as an intermediary between the DNA of the chromosome and the amino acids present in the cytoplasm and plays a vital role in the synthesis of proteins from amino acids. Some forms of RNA are confined to nuclei. The small nuclear RNAs, snRNA, are concerned with RNA splicing, page synthesis of protein. We have seen that a protein is made up of amino acids that are linked together in a definite sequence. This sequence is determined by the order in which the bases are arranged in a strand of DNA. Each amino acid is represented in the DNA molecule by a sequence of 3 bases, triplet cone. It has been mentioned earlier that there are 4 bases in all in DNA, namely adenine, cytosine, thymine, and guanine. These are like letters in a word. They can be arranged in various combinations so that as many as 64 code words can be formed from these 4 bases. There are only about 20 amino acids that have to be coded for so that each amino acid has more than one code. The code words for some amino acids are shown in Fig 1.41. The code for a complete polypeptide chain is formed when the codes for its constituent amino acids are arranged in proper sequence. That part of the DNA molecule that bears the code for a complete polypeptide chain constitutes a structural gene or cistern. At this stage it must be emphasized that a chromosome is very long and thread-like. Only short lengths of the fiber are involved in protein synthesis at a particular time. The main steps in the synthesis of a protein may now be summarized as follows, Fig. 1.44. The 2 strands of a DNA fiber separate from each other, over the area bearing a particular cistern, so that the ends of the bases that were linked to the opposite strand are now free. A molecule of messenger RNA is synthesized using one DNA strand as a guide, or template, in such a way that one guanine base is formed opposite each cytosine base of the DNA strand, cytosine is formed opposite guanine, adenine is formed opposite thymine, and uracil is formed opposite adenine. In this way the code for the sequence in which amino acids are to be linked is passed on from DNA of the chromosome to messenger RNA. This process is called transcription. Transcription takes place under the influence of the enzyme RNA polymerase. That part of the messenger RNA strand that bears the code for one amino acid is called a codon. This molecule of messenger RNA now separates from the DNA strand and moves from the nucleus to the cytoplasm, passing through a nuclear pore. In the cytoplasm the messenger RNA becomes attached to a ribosome. As mentioned earlier, the cytoplasm also contains another form of RNA called Transferrna. In fact there are about 20 different types of Transferrna, each corresponding to one amino acid. On one side Transferrna becomes attached to an amino acid. On the other side it bears a code of 3 bases, anticodon, that are complementary to the bases coding for its amino acid on messenger RNA. Under the influence of the ribosome several units of Transferrna, along with their amino acids, become arranged alongside the strand of messenger RNA in the sequence determined by the code on messenger RNA. This process is called translation. The amino acids now become linked to each other to form a polypeptide chain. From the above it will be clear that the amino acids are linked up exactly in the order in which their codes are arranged on messenger RNA, which in turn is based on the code on the DNA molecule, but also see below. Chains of amino acids formed in this way constitute polypeptide chains. Proteins are formed by union of polypeptide chains. The flow of information from DNA to RNA and finally to protein has been described as the central dogma of molecular biology. Some further details about genes and protein synthesis. In addition to the protein coding sequences of bases, DNA also bears other regions that have a controlling function. These regions provide signals for initiation and termination of the process of transcription, or for the control of the process in other ways. The DNA sequence that provides the signal for initiation of transcription is called the promoter. Binding of RNA polymerase to the promoter causes the DNA fiber to uncoil, and thus makes it possible for RNA polymerase to reach the fiber and to begin the process of transcription. Transcription continues up to the region of the DNA fiber that bears a code that gives a signal for termination of transcription. Apart from initiating the process of transcription, the promoter also determines the rate of transcription. Although all cells contain a complete complement of DNA, AII of it is not used for transcription in every cell. The region of DNA that is to be transcribed, in a particular cell, is determined by gene regulatory proteins present in the nucleus. These proteins bind to sites on DNA that are called enhancers. This binding is necessary before transcription can take place. Enhancers also control the rate of transcription by determining the number of RNA polymerase molecules that are engaged in transcription of the same length of DNA. Apart from sites on DNA that act as enhancers, there are others that act as repressors. Messenger RNA formed as described above is modified before it is used for protein synthesis. Long chains of mRNA are broken up into short lengths. This process is called splicing. Some of the pieces formed by splicing again join together, in modified sequence, to form a new chain that is used for protein synthesis. Other short lengths are destroyed. Combination of the short lengths in different ways can enable formation of different proteins under the influence of the same DNA sequences. It will be obvious that only some of the DNA sequences used for transcription will actually be used for protein synthesis. These sequences are referred to as exons. Those not used are called introns. The process of RNA splicing may be regarded as a method to get rid of introns. Why introns are created in the first place is not clear, but they probably perform some regulatory function. The initially formed mRNA, or primary transcript, is modified as follows. A. The mRNA has two ends. The end at which transcription begins is the 5' end, the other being the 3' end. A molecule of methylguanine gets attached to the 5' end, and is called the methylguanine cap. The cap is responsible for protecting mRNA from degradation. The opposite, 3' end, is referred to as the tail. This tail is also concerned with stability of mRNA. B. As mentioned above, the process of removal of introns from the primary transcript is called splicing. Splicing takes place while mRNA is still within the nucleus. The mechanism of splicing is complex and we will not consider it here. Apart from removal of introns, splicing allows exons to be united in sequences different from the original. C. After entry into cytoplasm, most mRNAs have a short life, a few minutes to a day, after which they are destroyed. The first region to undergo destruction is the tail, and this exposes the rest of mRNA to the action of ribonuclease, which is responsible for destruction of mRNA. Formation of proteins by translation of mRNA is controlled by various factors. Newly synthesized protein often needs modification before it is in its final form. The modifications may include a process of folding which may require the presence of accessory proteins. b. Addition of other molecules, sugar, phosphate. c. Cleavage of the originally formed protein to generate an active form. Role of ribosomes in protein synthesis. Ribosomes play an essential part in protein synthesis. They read the code on mRNA and help to arrange units of tRNA in proper sequence. The two subunits of ribosomes, large and small, play different roles in protein synthesis. The smaller unit, 40s unit, is concerned with the process of translation. The larger unit, 60s unit, is responsible for release of new protein into the vacuoplasm within the cisternae of endoplasm IC reticulum. Messenger RNA entering the cytosol meets a free ribosome. Messenger RNA has an initial part that bears the code for a signal sequence. This signal sequence tells the ribosome about the nature of the protein to be formed, and determines how the ribosome will behave. A. If the signal is for a protein that is to remain in the cytosol, the ribosome does not get attached to ER. The protein synthesized is released into the cytosol. B. If the signal is for a membrane protein, or for a protein to be secreted, the ribosome attaches to the surface of ER. Membrane proteins synthesized get incorporated into ER membrane. They are transferred from rough ER to smooth ER, thereafter, to the Golgi complex, and then to the cell surface. Proteins that are to form secretions enter the lumen of rough ER. They pass into the lumen of smooth ER, and then, through vesicles, to the cis face of the Golgi complex. After being appropriately processed in the Golgi complex, they are packaged into vesicles and are discharged from the cell by exocytosis. Duplication of chromosomes. One of the most remarkable properties of chromosomes is that they are able to duplicate themselves. From the foregoing discussion on the structure of chromosomes, it is clear that duplication of chromosomes involves the duplication, or replication, of DNA. This takes place as follows, Fig 1.45. The two strands of the DNA molecule to be duplicated unwind and separate from each other so that their bases are free. A new strand is now synthesized opposite each original strand of DNA in such a way that adenine is formed opposite thymine, guanine is formed opposite cytosine, and vice versa. This new strand becomes linked to the original strand of DNA to form a new molecule. As the same process has taken place in relation to each of the two original strands, we now have two complete molecules of DNA. LT will be noted that each molecule has one strand that belonged to the original molecule and one strand that is new. LT will also be noted that the two molecules formed are identical to the original molecule. Structure of fully formed chromosomes. Each chromosome consists of two parallel rod-like elements that are called chromatids, Fig 1.46. The two chromatids are joined to each other at a narrow area that is light staining and is called the centromere, or kinetochore. In this region the chromatin of each chromatid is most highly coiled and, therefore, appears to be thinnest. The chromatids appear to be constricted here, and this region is called the primary constriction. Typically the centromere is not midway between the two ends of the chromatids, but somewhat towards one end. As a result, each chromatid can be said to have a long arm and a short arm. Such chromosomes are described as being submetacentric, when the two arms are only slightly different in length, or as acrocentric, when the difference is marked, Fig 1.47. In some chromosomes the two arms are of equal length, such chromosomes are described as metacentric. Finally, in some chromosomes the centromere may lie at one end, such a chromosome is described as elocentric. Differences in the total length of chromosomes, and in the position of the centromere are important factors in distinguishing individual chromosomes from each other. Additional help in identification is obtained by the presence in some chromosomes of secondary constrictions. Such constrictions lie near one end of the chromatid. The part of the chromatid distal to the constriction may appear to be a rounded body almost separate from the rest of the chromatid. Such regions are called satellite bodies. Secondary constrictions are concerned with the formation of nucleoli and are, therefore, called nucleolar organizing centers. Considerable help in identification of individual chromosomes is also obtained by the use of special staining procedures by which each chromatid can be seen to consist of a number of dark and light staining transverse bands. Typically the centromere is not midway between the two ends of the chromatids, but somewhat towards one end. As a result, each chromatid can be said to have a long arm and a short arm. Such chromosomes are described as being submetacentric, when the two arms are only slightly different in length, or as acrocentric, when the difference is marked, Fig 1.47. In some chromosomes the two arms are of equal length, such chromosomes are described as metacentric. Finally, in some chromosomes the centromere may lie at one end, such a chromosome is described as elocentric. We have noted that chromosomes are distinguishable only during mitosis. In the interphase, between successive mitoses, the chromosomes elongate and assume the form of long threads. These threads are called chromonemata, singular equals chromonema.